Thank you, and good evening. And thank you for the invitation to come and talk to Liverpool Geological Society. So, bearing in mind that I'm not a geologist, I have some expertise in image analysis. I have applied that image analysis to earth materials, which is why I'm talking to you this evening. So, there's a little bit of microscopy of earth materials at the end, but most of the first half, possibly even the first 2/3 of this talk, not about geology per se, it's about how you look at images. As was said in the introduction, I am interested in all aspects of imaging, earth materials being one of many. So the first question you might ask is, why should any geologist be interested in image analysis? Well, there's one overriding reason, and that is I want to sell a book. And therefore, geologists ought to be interested in image analysis. My interest in looking at images goes back a long, long time. And a little while ago, so a few years ago, I collaborated with Professor René Heilbronner at the University of Basel, and we produced this book. I have quite an extensive history of image analysis. Professor Heilbronner is a respected geologist, and we got together and put this book together. So, I'm going to try and convince you in the next few minutes that image analysis is something that geologists ought to think about. I'm going to give you a little bit of an introduction to image processing and image analysis, what the difference is and what we're talking about. And then I'll take a little diversion about the nature of light, simply because a lot of earth materials are studied by their interaction with light, either by photographing them in light or using microscopy and shining light through thin sections of samples in order to glean some information about the structure. So I'm going to say a little bit about the nature of light and basically say a little bit about what these demonstrations are about. And then I'll finish with how we actually carry out and what we do with microscopy of earth materials. And I invite you at the end to come down and have a play with a couple of the demos I've got lined up here, one of which is explicitly about how we go about looking at microscopy of thin sections, and the other one is just a little bit of a play with polarization and a calcite crystal. So that's what I'm going to be doing. In the next 3 or 4 hours, I'm going to take you through these various topics and see how we go. So firstly, why image analysis? Well, just very briefly, for instance, if we take a number of different samples, I may fall into the parlance of calling them earth materials or geomaterials. Occasionally I just call them rocks, because that's all a physicist really understands. Some rocks you can basically say are essentially particles in a matrix. Here we see what appear to be almost circular particles, and we can reduce that image to simply white particles on a black background. So we have two types of interesting thing in this image. We have the particles themselves, not necessarily all the same size, not necessarily all the same shape, and they're sitting in some sort of a background matrix. And that's different from the aggregate in the middle, where again we have particles or grains of the particular mineral in question. But here, instead of having large gaps in between so that we can clearly see the shape of these grains and gaps in between them, here they're all touching. The aggregate means that all of the grains, small and large, are effectively producing, again, rather ragged looking shapes, rather more than the rounded shapes we can see on the left-hand side, but they all basically interlock together. So here the black is actually a medium, and it occupies a certain amount of space, a certain area, a certain volume, whereas here the black lines simply delineate the boundaries between one grain and another. It's not a different medium per se. And in the third category, we can see that we can't make out individual particles. We can't see where one grain touches another grain. We can get the impression of a texture in the material, but we can't identify, if you reduce that image to black and white, as we have done the previous two, you can't see individual grains of the rock structure. So you can see that if we want to understand what this rock is, what its history is, how it's formed, how it changed with temperature, how it changed with pressure, how it's come to be the way it is, then we need to understand something about these various structures. Put them all together, we can simply label them as the microstructure of the material, the microstructure of earth material, meaning the microscopic scaled structure. And you can see these are very different, so we would expect them to behave differently, and we would expect them to have different origins. And in terms of understanding not only the structure, we would of course need to understand the structure in the context of how these particular earth materials or rocks came to be. Again, I am not making any particular comment, it's just what little I know about geology. I assume that is the projector giving that warning? Okay, just goes away, does it? So for instance, we might have a cataclastic rock which is formed through processes such as fracturing and crushing, and we can perhaps see that, and we can get an impression that we have grains in there of various sizes from huge to tiny, and we can get an idea of the history of that particular sample by looking again and analysing the distribution of, for instance, size and shape of the various grains within the structure, and that tells us something about the fracturing and crushing and the forces involved that produce that particular type of rock. And here in the middle we have a marble. I'm going to say a little later about where the colours come from. These are not colours in the sense that we don't shine a light on the material and then that's the colour we see. I'll say where these colours come from. But here we have a recrystallised marble, and the crystallisation and the sizes of the grains will depend on how that marble has been subject to stress and strain, either its immediate stress and strain or its history of stress and strain. So if we wanted to understand the physical processes, we need to analyse what we have actually got there. What are we actually looking at? What size of grains, what distribution of grain sizes? And a third different category, oolithic or oolitic limestone, where we're looking at individual oolites there, where studying what we have in terms of structure or structure can give us information about the deposition, how that rock came to be because of the deposition of the oolites, I don't know how many years ago. So again, if we want to understand physical processes, we need to understand how those physical processes affect the structure, and we need some way of quantifying that structure. If we don't quantify the structure, then geology isn't a science. It's just, ooh, look at that pretty rock. If we want to actually make it a science, we've got to actually quantify what it is we're looking at in terms of the structures, and once we've quantified structures, we can get an idea of physical processes. So that is why we need image analysis and image processing. So firstly, just the distinction between the two. Of course, they're both starting from the point of images, but what are we actually talking about? Two separate concepts, but related. Image processing takes one image and turns it into another image. It does something to the image, hopefully, to make some aspect of that image more readily interpretable, usually by eye, sometimes by computer. Image analysis means quantifying some aspect of the image. So, for instance, in the context of geomaterials, that might be finding out what the distribution of particle grain size is within the material. So image processing, one image to another, image analysis, getting some numbers out of the image. And I'm going to illustrate the concepts of processing and analysis to start with a few images outside of earth sciences, and then I'll segue into the earth sciences. So especially when we're dealing with digital systems, I'm going to assume all of this is digital. None of this is analog imaging of samples. No, take a photograph and then hold it up to the light. This is digital images. How are we going to analyze them with a computer? Images are just numbers. No matter what you think you're looking at, all you're really looking at is a collection of numbers. And we choose that those numbers are represented by usually shades of grey or possibly colours in order to produce an image which we readily recognise as a two-dimensional image, which our eyes and ultimately our brains can then process. But as far as the computer is concerned, the computer doesn't really give a damn. The computer just knows it's got a huge array of numbers to crunch, and that's how we do the image analysis. We can take an image. This happens to be a high-resolution image of individual, what is this? This is silicon. These are individual silicon atoms sitting on the surface. We can choose, we've got an image which is just a collection of numbers. We can choose to assign each number to a shade of grey, or we could give it an arbitrary colour. This is, of course, nothing to do with the actual colour of whatever it is we're trying to image. This is simply, given that we've got a whole load of numbers in the computer, we can just choose to label each number with a particular colour, and it might make the structure that we're trying to look at more clear than just applying a grey scale. And if we wanted to, we could instead apply, contours. There's no limit really in how we choose to actually display an image, i.e. display a collection of numbers. If we're dealing with a microscope that does height information as well as position in X and Y, then we can draw a three-dimensional surface. This is that same silicon surface, and the scale here you noticed is a few nanometers. So that's just a reminder that although we think of this as a window onto the sample, really it's just a collection of numbers which can be represented in grayscale, or depending on the resolution, we can either have high resolution or low resolution and a three-dimensional representation or a rather blocky representation, but they are all representations of the original data. If we have a sample that looks like that, unlike the case just shown where we arbitrarily can color the sample, in this particular case, I've lost it, oh here we are. If we take a thin enough section of rock, the rock sample will basically just be transparent. There'll be absolutely nothing to see. So the colors here are not us arbitrarily deciding to apply a particular so-called color palette to the sample. These colors come from the process of imaging the sample, and I'll show you how that is done by a demonstration in a little while. But if we assume that's the starting point, an image containing certain grains of the sample, it is possible to process that image and generate another image, perhaps one that highlights the edges, the grain boundaries between the grains, and highlights the structure. In this particular case, we've processed it in such a way that what's going on inside the grains is essentially irrelevant, and only the network of grain boundaries is shown. Alternatively, or perhaps via one of these routes, we can actually analyze the image to say, well, how many grains do we have of a particular size, a particular radius, if you like, and then we can actually quantify. It is not unusual to do some image processing before we do the image analysis. That's not a requirement, but processing leads to another image, analysis leads to some collection of numbers, depending on what it is we're trying to get out of the sample. So if we want a computer to do this, generally speaking, we need some software to do it. And I started getting interested in image analysis is actually writing software in order to analyze images so that myself and my students could actually take an image and then do something with it to extract meaningful information. So there's me a few years back with a temperature warning apparently. There's me in front of a microscope. This particular microscope is a scanning tunneling microscope. It's not one that's necessarily used for earth materials very often, but it does have very high resolution. It involves moving a very sharp tip over a surface. It's only a tiny distance, nanometers or so, just a thousandth of a micron, a millionth of a millimeter if you prefer, a billionth of a meter if you prefer, only a nanometer or so above the surface, and through a quantum mechanical process I'm not going to go into, it allows us to get individual resolution of atoms on a surface. This is the surface of rust. You can tell it's rust from the color, and the individual atoms of iron and oxygen can be resolved. You see the scale down here of 2 nanometers. This is possibly one of the highest resolution images you can get. No, whoops, sorry. Notice that the scale here, 850 picometers. So this particular image is less than 1 nanometer by 1 nanometer, 1 billionth of a meter. If we wanted to image, for instance, a grain of sand, on this scale, a grain of sand would be about the size of the moon. That gives you an idea of how much we've zoomed in this particular case. I'm telling you this because this is I started writing software to analyze images that were coming from tunneling microscopes. And I was interested in the atomic arrangement. How are atoms arranged on the surface of various materials? My main interest was in a certain set of metals called the rare earth metals, but starting by imaging atoms, I then later realized, well, the same software could be used for analyzing images not from scanning tunneling microscopes, but from scanning electron microscopes or from microscopes. And there's no reason why you should think of the software as only being good for looking at atoms on a surface or molecules on a surface. It could look at a whole load of other things. So I did some imaging. This is a scanning tunneling microscope of some of these metals, these rare earth metal surfaces. That's where I cut my teeth in the sort of 1990s, as it were. And here's an example of what you can do with a little bit of image processing. If we wanted to process that image to make it a little clearer, this is not the best image in the world. I'm deliberately using a rather poor one rather than trying to get a better image. For the sakes of demonstration, this is a rather poor image of a lot of different atoms on the surface of a metal alloy. But if we do some image processing, and we can do what's called Fourier filtering. If you don't know Fourier analysis, don't worry. It's simply a technique whereby we can actually take the image, and by processing that image, we can actually end up with an image of the same size and the same resolution, which hopefully shows us where these atoms are in much more detail than we can see in the original image. It's almost to the point of suspicion where you think, how on earth can you get anything so clear out of anything that's so grubby? Well, you've got to be careful that you're not over-processing the image, and you've got to be careful you're not fooling yourself into thinking that things are real. But as a check, for instance, we can look at a little rosette of atoms that form this tenfold ring here, and if we look back to the original image, it's rather more fuzzy, but I hope you can see from where you're sitting that there is actually a tenfold little flower there, just like there is there. It's simply in the process image, it is made so much more clear, high contrast and a lot clearer and a lot less noise. We're able to interpret that one more easily. So image processing helps us interpret what's going on, and the actual way we get there actually helps us with the analysis as well. Here's another example where we're interested in molecules sitting on a surface. Each of these little triangle things is a little molecule, and the molecule can sit in 1 of 4 different orientations on the surface, indicated by these symbols down the left-hand side. That's what we would expect the molecule to look like. And I wrote software to try and automatically pull out and recognize which particular orientation are each of these molecules. The molecules are only a each, so it's a little bit tricky, but ultimately we can convert a real picture of the sample to, if you like, a stylized or sanitized version where we're just indicating what molecules are present and which way up they are. The interest was, will we find as many red as blue when we do this analysis? How do the molecules decide which way round they want to go on a surface? In the end, it looks like, well, perhaps they're randomly distributed. It's not like they are in any particular regular array. And I was also interested in, can we actually identify where molecules sit? They seem to be on a sort of grid in this particular system. They're almost on a regular grid, but all the molecules seem to be nudging each other and pushing each other out of the way, such that none of them is quite on the grid that you would expect. And image processing allows us to find where the atoms are, and in this particular case, I colored each little blob with how far away from where you would expect it to be is it. In other words, the dark blue molecules are exactly where you would expect them to be if they were on this regularly grid. But if the atoms are getting nudged away from that grid, they get labeled a lighter green or orange or eventually red. So this is just a way of visualizing, helping us interpret. So this is sort of a hybrid, if you like, between image processing and image analysis, because we try to analyze where the molecules are and then represent it in a colored way such that we can more easily look at that and get a picture of where the molecules are compared to the original image of molecules. So that's a little bit of background of where I started writing software to do image processing and image analysis from various microscopes. Let me take one of 2 asides and say a little bit about visual perception. Because a number of people, including, no disrespect, but including biologists who often look down microscopes at various messy samples like biological squishy stuff and try and figure out what's going on by looking at a sample, looking at a specimen of some kind, and using their eye and their brain to figure out what's happening. Now, I agree, the eye and the brain, an amazing combination for analysis, but eyes and brains can be fooled, and sometimes we have to put that aside and say, no, we must have the computer make the decision, not an eye and a brain. So, for instance, the main problem I will show in a couple of slides, and the problem is what we see isn't always what's actually there. In other words, we see what we think we see. Your brain is taking an awful lot of shortcuts. In some sense, your brain is doing an awful lot of image processing whilst you're looking at an image, trying to figure out what it actually is. So you can ask, well, which is the better image processor? Is it a brain or is it a computer? Computers can do some things your brain can't, and your brain can do some things that computers can't. Is it better to analyze or process images using a brain made of carbon, i.e. us, or a different type of brain made out of silicon, i.e. a standard electronic computer? Well, the answer is sometimes one and sometimes the other, but I'm going to try and convince you that the brain and the carbon side of things can be very easily fooled. So, for instance, have a look at this. We've got a cube. You may have seen this before. Anybody colorblind? Okay, sorry, sir, it's not going to work for you. Okay, for everybody else in the audience, there's a cube on the left, and there's a cube on the right, and I'm interested in the colors on the top of the cube. Forget what's on the two faces. So can you see on the top of this cube some tiles that are blue? How many? I think everybody said 4, okay? Take the cube on the right. How many of those tiles on the cube on the right are yellow? Yep, I think everybody said 7. So does that mean you're all happy that on the left there are 4 blue tiles and on the right there are 7 yellow tiles on the tops of those cubes, yes? Would you put money on that? If so, how much? If you rely on your sense of color blindness notwithstanding, you can be fooled. So although it seems perfectly obvious that there are 4 blue tiles on the left and yellow tiles on the right, you may be interested to know that those 4 tiles that you've labeled blue in your head and those 7 tiles on the right which you've labeled yellow are all the same color. All 11 of those tiles, the ones you think are blue on the left, the ones you think are yellow on the right, they are all the same color. And you can either believe me or not. You can go to the relevant web page and do the test yourself if you don't believe me, if you think I'm hoodwinking you. But one thing I can do is remove all of the pixels other than the ones inside those 4 tiles and inside those 7 tiles. So I'm going to take away all the other pixels so you know what you've actually been looking at. And here they go, one at a time. Whoop. Sorry, just went too far. Those are the tiles you were actually looking at. You thought those 4 were blue. You thought those 7 were yellow. Only because of the other pixels that happen to be in the neighborhood. If I put some of the colors back in, you notice that those blue are surrounded by a wash of yellow, and those yellow are surrounded by a wash of blue. Your eye, or explicitly your brain, can be fooled. And your brain is simply telling you, well, that's gray, but it's bluer than other things in the neighborhood. And your brain is telling you, well, maybe that's gray, but it's more yellow than other things in the neighborhood. And now you know that these aren't blue and these aren't yellow. Can you force your brain to convince you? There is actually a little trick you can do, and that's you just have to block out the other pixels. I did it electronically. You can do it yourself if you go to the same web page. Just get a sheet of paper and poke a pencil in such that you can only see the pixels from one area. You can almost do it yourself if you just make a very small, I'm not sure you can make a hole small enough, maybe on the front row, but probably not on the back row. You can't make a hole small enough so you can only see the pixels in one of those tiles. If you could, you would then say, hey, the blue's gone, it looks grey. And if you do that over here, hey, look, the yellow's gone, it now looks grey. It's easier to do on a computer monitor where you're sitting right in front of it. If you go to that web page and find this particular cube, there's lots of optical illusions on that page. But this, I thought, was quite dramatic, and it convinces me absolutely that if you're looking down a microscope and you're trying to recognize, the parasite I'm looking for, if it's grey, that's a cell, but if it's blue, that's a parasite. You cannot rely on that as a way of doing science. When I told a postdoc in biology, he was rather horrified because that's precisely what he'd been doing for the last 6 months. And then he realized that his sense of color judgment could not be relied on as a scientific method. And if you think it's only, let's go back to the end again, if you think it's only color rather than anything more straightforward, then, well, no. Here's another optical illusion that you may have seen before. I'm interested in the square with an A on it, not the A itself, the square on which the A sits, and this square here on which the letter B sits. I'm interested in the relative brightness of A and B. So if you're looking at a rock sample or looking at an image of a rock sample and one grain appears to be twice as bright as the other gray, is that something you can rely on in terms of your judgment when looking at an image? So if asked, perhaps it's not a fair question, but if asked how much brighter is that square than that square, people might say, well, it's a factor of 2 brighter, or maybe a factor of 3 or something like that. But all we have to do is block out some of the additional pixels on the right and on the left. So what are the actual brightnesses of square A and square B? You can see that A is actually exactly the same gray as the gray to the left or the right, and B is exactly the same gray as to the left and the right. There is no difference between that gray and that gray. A and B are on squares that are exactly the same shade of gray. It's not just roughly the same. I've checked it. They are exactly the same shade of gray. And so if we take those away again, what is your brain telling you? You know that they're the same. You know they're the same. I've just showed you they're the same. And yet your brain is still telling you that the A is on a darker square than the B. So not only can you be fooled, there are times when you can't even train your brain to recognize what it should be seeing. So that's why you've got to be careful when you're looking at images. It is far safer if you're interested in what is this color or what is this brightness or shade of gray. It is far, far safer to let the computer tell you what that number is rather than you say, ah, well, I can see they're the same. If you're looking at an image and you want to tell if things are the same, you think your brain are good at doing this. In fact, they're not. They can easily be fooled. And here's one example of something where it looks easy, but it actually was quite tricky. A colleague was interested in measuring the positions of these wiggles, which are DNA strands. The DNA strands on a glass background, and you look at that and you think, well, they're easy to see. I can see where they all are, no problem at all. And I assume that if I were to take a slice through here and look at the brightness, or height in this case, as a function of position, surely it would look something like that. It would be a little bit of background, a little bit of noise, and then I would perhaps hit one of these things, which obviously is a lot brighter, and so it would be a lot higher in this image. So if just ignore the nanometers, just think of this as brightness. So clearly there's a bit of background, then every so often there's a DNA strand, a bit of background, a DNA strand, etc. So if I were to slice through, every time I cut through a DNA strand, I would see one of these things, presumably. Well, actually, no. What you think is there, but what is actually there is much more like that. That is the background and the DNA. The slice I've taken is this slice here. And so you can see that there's nothing, 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 and then there's DNA there, and then the next slice of DNA is there, and then the third one is there. That corresponds to these 3 points. So those 3 are the obviously bright objects, the worms, the DNA strands that are on the surface. But if you ask a computer to pull them out, well, they're not the 3 highest points. You can see there, okay, the highest one happens to be DNA, but the second highest isn't a piece of DNA. The third highest isn't a piece of DNA. This is one example where what the computer sees and what you see are very different things. Your eye and brain find it very easy to pick out where these wiggles are. The computer, if simply asked to pick out the highest points, would not pick out the DNA. It would pick out a whole lot of rubbish in the background as well. So in this particular case, software had to be developed which allowed the image to be processed in such a way that the DNA could be in your face obvious, rather than I think I can see all of these things. The computer has now processed the image such that it is, if you'll excuse the pun, absolutely black and white as to what is DNA and what is background. So brain versus computer, you've got to be careful, is the bottom line. Before I talk about how you can image Earth materials, I just need to take a second diversion into the nature of light, because it matters. If we are going to be using microscopes, such as a light microscope, to look at Earth material, then we have to understand how that material is interacting with light, how light is interacting with the material. So, electromagnetic waves, basically visible light would be the same if we were talking about ultraviolet or any electromagnetic wave, but basically we have an electromagnetic wave in which a magnetic field and an electric field, they're oscillating at 90 degrees to each other. That doesn't really matter so much, but the direction of propagation of the light is such that the electric and magnetic fields are varying backwards and forwards at right angles to the direction that the light is traveling. And so we have this concept of polarization when it comes to looking at light that goes wiggling its way through life. We think about electric and magnetic fields, and we know it's electromagnetic, so we know there's an electric and a magnetic field, but for convenience, physicists will never draw, except in this particular example, never actually draw the magnetic field. We know it's there. We know it's at 90 degrees to the electric field. So when we think of a light wave, we just think of what is the electric field doing. The electric field is bobbing up and down, for instance. But of course, it doesn't have to bob up and down that way. It could go from side to side. So we don't worry about the magnetic field. In other words, take away all the purple and just think about the green. Then we have possible polarizations of the light traveling, in this particular case, from your left to your right. The polarization can be such that the electric field is vertical, or the polarization can be such that the electric field is horizontal. Wait, I hear you ask, can't it be something in between? Well, yes, but that's just mathematically a combination of the two. So we don't need to worry about what if it is 45 degrees or what if it's a little further over. We just think of how much of the field is polarized that way, how much of the field is polarized that way. There are other types of polarization of light. It is possible, I'm not going to give a demonstration here, that's part of a different talk entirely. It is possible for light to end up state, which is not simply wiggling up and down or wiggling side to side. It's possible to generate light which is actually corkscrewing in the clockwise sense or corkscrewing in the anti-clockwise sense. You might have heard of these: left polarized light and right polarized light, or left circular polarized light, right circular polarized light. So I'm not going to spend much time talking about this. I am interested in the polarization because of what we're going to see with a little demonstration here. So first, let me show you what it is I hope is going to be shown here. We're going to take a polarizer, a polarizing filter, just like a Polaroid sunglasses, that only lets through light if it's wiggling side to side, if it's wiggling horizontally. And we're going to take another sheet which will only let through light if the polarization is vertical. Okay, so that's what the arrows represent, only letting through light if the polarization is horizontal, i.e. the electric field is doing this, forget the magnetic field, and this sheet will only let through light if it's vertical. So if we put the two sheets together, no light can come through because light can't be that vertical and horizontal at the same time. It's one or the other. So let's see if we can actually demonstrate this. What I'm going to do is put two polarizers together and show you that no light gets through when you have this situation. And then I'm going to show you what happens if we take another piece of plastic, a completely transparent piece of plastic. What if we put that piece of plastic in between the two polarizers? So that's what I am going to do now. So let's see if this is still working. Okay, I've now switched to showing you what the phone is seeing. So let me switch the light on. Okay, so there's a rectangular light. And here's two pieces of Polaroid. One piece of Polaroid doesn't show you anything in particular. It's sort of most of the light gets through. The other Polaroid, most of the light gets through. But if I put them together and turn them around such that they are in the configuration I showed on the previous slide, whoops, let's see if I can do this without getting it in the wrong place, you notice the light goes away. So now we're in a position where no light is getting through. Let me just pull it across a little bit. That's a bit better. So apart from a few specks which it's having difficulty focusing on, no light is getting through the polarizer. What if I now take my sellotape, which I have now very conveniently lost. What did I do with my... Haha, I put it down just a second ago, and I now can't remember where I put it. Being transparent, it doesn't always help. Just give me a second. I can't remember now if I put it down here or... Oops. Found it by sound rather than by feel. So we have two polarizers here. And so no light is getting through because the first one is blocking horizontal, the second one is blocking vertical. So what difference could it possibly make if I add a piece of plastic in between them? This particular piece of plastic is transparent plastic with some transparent sticky tape on it. What happens if I introduce the plastic in between the polarizers? There we go. So you see what we see? Not only do we now see light, compared to if I take the plastic away, there's nothing, but if I put the plastic in, light comes through. But not only that, but we get colors showing through. And different colors, you can see, are different thicknesses of sticky tape. You can see the edges of some pieces of tape there, and if there's multiple pieces present, you get different colors. And if I rotate the sellotape, you can see that the colors change. So we're getting colors. It's nothing to do with the sellotape itself. Let me just show you what the sellotape looks like. That's it. You can see a few bits of tape there, looking a little bit messy, but completely and utterly colorless, just like a rock sample would be. If you took a rock sample and thinned it down to a few microns, there would be essentially no color in the sample. But if you put that rock sample between two polarizers and then put that into a microscope, then you will see essentially the structure of the rock sample, because you will see different grains reflected in different colors in the image. Let me just show you what's actually going on there. Let me kill this and go back to the PowerPoint. So what's actually happening, that's what we effectively saw, perhaps not as contrasty as what you saw a moment ago, but here's the actual plastic itself, sellotape, which is essentially transparent. But if I put that sellotape between two polarizers, now we see that not only does some light come through, but the light that comes through is colored simply because the process by which we are taking light with lots of different polarizations. That lamp has no particular polarization. Some light is coming out vertical, some light is coming out horizontal. We have one polarizer which only lets through light, let's say vertically, and then you have another one here. Sorry, this is actually arranged in a slightly different arrangement. But what happens to the light that comes through is when we go through a piece of sellotape, just like a lot of rocks, is what we call optically active. It will change the orientation of the polarization. In other words, any light which is coming through, let's say, the vertical, if it comes through the vertical, before it goes through the horizontal, the sellotape or the rock sample is changing the polarization. The light is coming through vertical, but the sellotape or the rock is actually changing it so that some of that light will make it through the next polarizer, which would be horizontal in this particular case. And the amount of light that gets through depends on how much it's changed, in other words, how thick this material is, hence the different thicknesses of sellotape, each of which give a slightly different rotation of the polarization, and light of a particular wavelength will be rotated by a certain amount and will result in certain colors being present for certain thicknesses of the sellotape. Another example of optical activity, which I invite you to come and down and have a play with. It's difficult to actually demonstrate on a projector, as it were, but basically I've brought along a piece of calcite, and if you use calcite, you've probably seen this before, this so-called double refraction. If you put a calcite crystal down, you see two images. Those two images are different polarizations. So I brought along a piece of calcite and a polarizing filter. So if you look through the polarizing filter at the crosses, you will see something interesting, which I will wait until the actual demonstration itself to demonstrate. So that's how microscopes work. If we're interested in looking at rock samples, that's why we use polarizers. That's why virtually every optical microscope that looks at rock samples is some sort of a polarizing microscope, because doing that, instead of getting a thoroughly boring image that we can hardly see what on earth is going on, we can barely make out what's there, we can hardly make out any grain boundaries at all. As soon as we put it between polarizers, then we get visible grains within the sample. So let's just have a quick look at how we might image a piece of rock. One option is simply polish the sample and then scan it, or possibly with an acetate foil, press that to the surface and then peel that off and image that. Taking the sample onto an acetate foil first has the advantage that you get a genuinely two-dimensional image of the sample. In the case of this, for instance, although you think of it as a surface, you polish a surface and then scan it, but if the matrix of a whole load of particles is transparent, you are actually looking into the sample, perhaps only by tens or hundreds of microns, or maybe a fraction of a millimeter, but you are looking into and hence making a three-dimensional judgment of what the structure is. So in that sense, an analysis of a foil impression of the surface is actually a better two-dimensional indication of what that sample looks like. So for instance, if we're looking at this oolitic limestone, we're looking at the individual oolites, but we can see gaps which are actually transparent matrix, and we're looking further into the sample. We don't have that problem if we look at the scan of an acetate foil, because that gives us a genuine two-dimensional indication of what's happening only at the surface, not what is happening just below the surface. And of course, we can take thin sections. We can take sections only a few microns thick, and then we can actually, again, put them into a microscope so that we can see the structure. And here we're still talking about relatively macroscopic, so we're talking about millimeters here rather than microns in terms of the structure we're interested in. But here is an indication of what you might see if it was just a thin section was imaged just with a light microscope and no other filters. We might get the impression of edges of grains, just like we can just about make out individual bits of sellotape stuck to a piece of plastic. We can just about get the indication of a ghostly form of the grain boundaries. Whoops! But if we want to see exactly what's going on, or in this case with C in the bottom left there, if we want to get different colors corresponding to different orientations of the grains within the sample, then we need to use polarizers. And again, to reiterate, those colors are natural in the sense that they are produced by the interaction of the material with light. They are not imposed by us choosing to actually label some of these things orange and some of them blue because we think that's a pretty way of labeling the difference between one grain and another. So for instance, with any image, we have that ability to start with a grayscale image where the intensities are displayed purely as a function of levels of gray, and we can choose to color that in any way we choose to try and bring out the difference between one level of gray and another. But these are totally artificial in that all we've done in the computer is to say, for a given shade of gray here, represent it by this color, or represent it by that color, or represent it by that color. Exactly the same structure, and just choosing a color scale that might, in some circumstances, make it a little easier to see what those grains are doing. That is not the same as this here, in which the colors are produced in the microscope by the way the rock interacts with light. So I'm trying to just make sure you understand the distinction between those two situations. Coming back to the idea of we want to understand structure, so for instance, if this is our limestone sample, if that's the three-dimensional sample, we might be interested in getting the computer to isolate all of the circular oolites in this particular sample. But that's rather difficult, not least because we have this three-dimensional problem of looking into the sample, and it's difficult to distinguish one oolite from the next. So that's the raw attempt to try and isolate individual oolites. In this case, we've had a little bit of a tidy up, but we still have a little bit of a problem with the three-dimensional sense of the sample. If we take an acetate foil, then we end up with something that perhaps doesn't look as good when we start to try and isolate individual oolites. But once we realize that we can just play around with the contrast and brightness of the image, you notice that it is now a lot cleaner and a lot easier to be able to see what the individual oolites are, what their size is, what their shape are from the original sample compared to the equivalent from the 3D sample. So you can see that there are benefits for using acetate foils for actually imaging the surfaces of various rocks. When it comes to separating what minerals are present in a given rock sample, here we have, I've forgotten actually what the original sample was, but this particular system contains some quartz, some plagioclase, some feldspar, some biotite, and some muscovite. And the way it's imaged, you can see that each of them produces a slightly different grayscale because of the way they're interacting with the light in the microscope. But remember, when we're looking at this, we should be careful. If we think that is one particular sample, but we think perhaps that's feldspar because we think it's the same gray as this one, remember we can't rely on our eyes to tell us this gray over here appears to be the same as that gray over there. For the reason that I showed earlier with that optical illusion, two things which appear to be different gray can be the same, and vice versa. Two grays that appear to be the same can be very different, and vice versa. So we rely on the computer to tell us, right, what is the intensity of this region here? Here we have these histograms which are telling us how many pixels vertically have a particular shade of gray, which is the horizontal axis here. And you can see that we have, for K feldspar, for instance, we have a whole load of pixels, but all centered around one particular value, all in the range 35 to 135 brightness levels. It doesn't matter what the units are in this case. It's simply a level of brightness that goes from 0 to 255. So biotite at this end, where's the biotite there? So this is the white end of the spectrum. All the biotite is up here at the white end. Feldspar is the light gray. Muscovite is next to it. Plagioclase is a little bit darker. Quartz a little bit darker still. So there are situations in which when we look at a sample, if we put the sample into a polarizing microscope, we get a different intensity of light coming out for each of the chemically distinct minerals in the sample, and then we can use the brightness to tell us what they actually are. And we can then take our original sample and we can say, right, we can separate out all the pixels that are white, and we can say, right, that's where the biotite is. All of these pixels that are black here are originally white here. Or take all the ones that were originally this particular shape. Whoops. Trying to connect to the cloud, very good. All of the ones that are a particular shade of gray, we can bring out and say that's where the feldspar is. Notice that it's having a little bit of a more difficult job with muscovite. None of these image analysis tools are perfect in the sense that we are looking at an image. The image might contain a little bit of noise, and therefore we're trying to pull out the identity of various pixels within the image using which shade of gray are they. Most of the time that works nicely. It works well for biotite, feldspar, reasonably well for quartz. For muscovite, there are also a few pixels up here which are very similar intensity to the pixels that we know that we've identified as muscovite, but perhaps are just mislabeled simply because by accident some pixels have the same shade of gray, the same intensity on coming out of the microscope. And it's not unusual to find a few pixels that simply refuse to be identified. So it goes a long way to be able to automatically say, here's an image, what have we got in that image? Your eyes tell you, roughly speaking, what's going on, but using the shade of gray, it is possible to segregate out, or so-called segment, the image into the various contributions. We might be interested, having identified a particular grain, we might be determined that, well, maybe that's a grain of marble. We're interested in what shape is that? we're not necessarily interested in just how much of the sample is made of this, but what do the grains look like? Are they elongated? Are they ragged? Are they elliptical? We can ask the computer to say how close to a nice smooth ellipse is this particular grain? Which way is it oriented? How long is it compared to its width? And which way is it oriented in the sample? Because that might tell us about the stress that the rock has been subject to if all of the grains have been pulled in one direction and tend to have an elongation in a common orientation. So again, that's something that computers are particularly good at taking a sample that sometimes is a little bit difficult to see what's going on, but once we've reduced the sample to a collection of pixels, whether they be black and white or an outline, then it's very easy to get the computer to analyze and start giving us numbers that are related to those grain structures. So for instance, if we have a slightly larger view, we have a number of grains in this sample. It's a little difficult by eye to see what's going on, but the computer can figure out that these are where the grain boundaries are, and therefore we have various grains within the system. It doesn't really matter whether we think of it as white on black or black on white. The computer doesn't care because the computer is just shuffling numbers. When it comes to working out what shape they are, we can't rely on the shape of any of these grains that's touching the edge, because, for instance, in this case, we've got no idea if the rest of the grain finishes it in a sort of circular manner or is a very elongated grain. So any grain that's touching the edge, we effectively have to throw away, but the ones that are wholly within the image, we can look at those and figure out, is there a preferential orientation to these particular grains? What shape are they? What size are they? What's the size distribution, and are they oriented in a particular way, which gives us a clue as to the formation of this particular system. That's one thing that computers are very good at. If we give a computer a whole load of what looks like the side of a Dalmatian, if we give it a whole load of objects, which might be grains in a rock sample, for instance, a computer will quite happily, within a fraction of a second, tell you that the first object has an area of this many square pixels, and is at position X and Y, and it looks like an ellipse with this length and this width oriented at this particular angle. That's the sort of thing that's very easy for computers to generate. And once you've got a whole load of numbers, then it's very easy to look at, for instance, size distributions. If particle number 1 has a particular size, and particle 2 has a size, and particle 3 has a size, whether or not it's a few dozen or a few hundred or a few thousand of these objects, it's then very easy to look at the size distribution to find how many have a particular size or how many have a particular area, or in this particular case, if this is a three-dimensional scan rather than a two-dimensional scan, how many have a given height as well. And having extracted that, we can look at an example like, for instance, what is this? This is Carrara marble, so it's either white or grey varieties, and we can look at the structure of that, and by eye we might say, well, white marble and grey marble seem to have quite similar sort of structures. If we ask for an analysis of where are the grain boundaries, and hence how large are the various grains in this sample compared to that sample, we find that we get white on the left and grey on the right. We get a somewhat larger sample that was blown up just so you can see them a little more clearly. We have something like 1,000 grains now rather than just a dozen or so. We have 1,000 grains in this white marble and 1,000 or so grains in the grey marble. Again, our eye isn't too bad at looking at that and saying, well, there appears to be a difference. Perhaps you can look at that and you can get an inkling as to whether the grains tend to be a bit larger or a bit smaller in one sample relative to the other. But the point is, we shouldn't be relying on our guesswork anyway. We let the computer tell us what the actual distributions are. And it turns out that it's not obvious perhaps, but the most common size is actually the same in both marbles. The most common size is, where are we, about 50 to 60, very similar. Most common, I'm looking at the wrong one. Most common is about 40 microns in diameter, but the average is a little bit different in the two cases. The most common is the same, but the average is about 60 microns in the case of the white and about 50 microns in the case of the grey. And that's something that's rather difficult to pull out of that sort of picture. You might perhaps be able to guess that one is larger than the other, but it's very difficult to be able to identify that the most common size is actually the same in the two, even though the distributions are slightly different. On the same scale, you can see the distribution of here goes to a maximum, and there's some grains all the way out to 150 microns, whereas in the case of the grey marble, it peaks at the same place, but it dies away. There's very few grains larger than about 120 microns. So the grain size distributions are different for these two types of marble. The bottom graphs are just the same thing expressed in areas rather than expressed in diameters of the grains that we can analyze. And coming back to how that tells us about processes, here there's a sample of quartzite that's being processed by compression, and as time goes by, the compression is changing the grain structure of the sample. So it starts at A, and with higher compression, we've got a change of the grain structure. Higher compression still, we go to the item C, and higher still, we go to D. So again, your eye, I'm sure, is telling you that it's changing, and it looks like we're going from large grains to small grains. But if you were trying to guess, for instance, what is the most common grain size, which is the modal value, which is the most popular grain size for all the grains in this image, and B and C and D, I'm sure you can pick out the fact that the grains appear to be getting smaller, but what is not obvious until you do the analysis is that actually the most common grain size actually is the same in all of them. That is very counterintuitive, but if you look at the distributions, the most common grain size is actually, what's that, 3 to 4 pixels or so, and in B it's 3 to 4 pixels, and in C it's 3 to 4 pixels, and in D it's 3 to 4 pixels. So the most common grain size is the same in each case, but clearly the distribution is changing. How many grains do we have as a function of grain size? The modal value is the same. We see there's a second bump. There's a lot of bigger pixels in the case of A, and that bump is going away, and you see the entire distribution is starting to sharpen up as we go to higher and higher compression. So yes, we are on average getting much smaller grain sizes because we're losing the large ones. That is very difficult to see just by looking at the original images. You can see things are changing, but you can't really hope to quantify how they're changing, how grain size changes as a function of compression, unless you do image analysis and pull out the grain structure and plot it out, and then you can see what's happening, and you can see that you've got, as it were, a bimodal distribution, two modes. That mode there seems to be disappearing as you apply compression, and it's pushing the entire distribution over to the left-hand side. But all the time, the most popular grain size is 3 to 4 pixels. By no means obvious when you look at it by eye. And again, to come back, this is not natural color in this particular case. We're not looking at the colors produced by a polarization microscope. Here, when we've got the grain structure, when we've extracted and analyzed the grain structure for white marble and gray marble, we can, if we wish, actually paint in the colors of the grains simply by size. Label the large ones red and the medium-sized one green and the small ones blue, simply because our color vision, except for the gentleman in the middle there, our color vision is such that that helps us understand the difference of these two. You can look at that and you can instantly see, even if you were defocusing your eyes, you can instantly see there's less red in that, telling you there's less large grains in this grey sample than there is in the white sample. And the amount of green and the amount of blue, it's very easy for our eye and brain to process that. So here we've colored the grain map simply to make it easier for us to look at the grain map and get an idea of why they're different. This is nothing to do with the colors generated by a polarization microscope. So let me finish by just saying that there are other ways that we can analyze images. My colleague, Professor Heilbronner, is particularly interested in this technique whereby a sample is presented to a polarizing microscope and the sample is rotated, and each grain, the let me make that a little clearer. The sample is rotated, an image is taken. The sample is rotated further, an image is taken. And every 10 degrees or so, the sample is rotated underneath the polarizers, which changes the intensities. Perhaps you can see that what is labeled here as grain number 3 starts off dark, and then as the sample is rotated, it becomes lighter and then goes back to dark again. A different grain starts off gray and then goes white and then goes dark. The variation of how a particular pixel or a particular grain changes as it's rotated in a polarization microscope will depend on the orientation of the atoms. We're back to thinking about atoms. We're not imaging the atoms, but the technique is sensitive to the alignment of atoms within the grain, because each alignment of a particular arrangement of atoms in a grain will result in a different variation of light, a different variation in brightness as the sample is rotated in a polarizing microscope. So by following how the light changes, dark to light, light to dark, etc., for each of these, we can actually analyze a whole set of these images taken in a polarizing microscope, and then we can actually analyze, not only we can see the grain structure, we can actually quantitatively say exactly which orientation the crystal structure is on this color map. Again, this is not a color map that's natural, it's not a color map that's arbitrary, it's a color map telling us that if we were to look at that grain there, for instance, that color purple corresponds to that point there, which tells us which particular direction the atoms are aligned in that grain. In other words, if they were pointing straight up, they would be white. If they're pointing that way, they would be green. If they're pointing the other way, they would be blue. If they're pointing this way, they would be cyan. If they're pointing that way, they would be red. So it's like pointing anywhere in an entire hemisphere, if you want to think of it that way. And so each of these, we've now quantified exactly which way they're pointing. We can see perhaps there are 2 grains which are very similar colors, but that tells us that they're oriented in similar but not the same direction. One of those grains has the atoms pointing this way, the other one has the atoms pointing in a similar but not quite the same direction, because they are similar but not quite the same colors. So by reducing the orientation, the atomic orientation of grains to one of these maps, we can get an enormous amount of information again about how that particular grain structure came to be, how it formed, the energetics involved, how the thermodynamics has affected that grain, how the stresses and strains and pressures of the history of that particular rock sample is reflected in the arrangement, the atomic arrangement of the grain structure that we can see. So we can see grain size, and we can see grain orientation, and we can see what the atoms are doing inside the grains. So this particular technique, I've labeled it Principia simply because it's usually called computer integrated polarization. It's often called KIP analysis. I wrote some software which I thought was, well, it's the principles of KIP image analysis. So I called it Principia as a shorthand, as it were. And this is a very powerful way of being able to analyze rock samples. It requires a reasonably sophisticated microscope. At the very least, you need to have the right polarizers present, such that when you rotate the sample, you can capture a lot of images, and then you can get the atomic orientation as well as the crystal grain orientation within a sample. So I think I've talked for over an hour now, so I think I'd better wrap up. So I hope I've convinced you that geologists, if you need image analysis, at least image analysis can be enormously useful to geologists to try and understand the structure, and in particular the microstructure of rock samples. I gave you a little bit of an introduction to image processing and image analysis, and a little bit about the nature of light, so that I could then introduce the idea of polarization microscopes before telling you a little bit about how we can actually use them. So now I invite you to come down. Not only could you pick up a handout with the slides that I've been talking about here, but if you wish, have a little play with the polarization demo and the calcite demo. Thank you all very much.